Let me tell you about the story of senescence. In the early 1900s, the cellular dogma was that if you take cells from an organism, such as a mouse, and put them in a petri dish, add optimal media, the cells would divide and grow indefinitely, making them essentially immortal. If you were to plot the cell count over time after each division, the cell count would be stable. However, in practice, cell cultures were unable to divide for more than a year, at which time they began to die. The inability to maintain cell lines indefinitely was explained away by either technical mistakes or suboptimal growth conditions that at the time couldn't be accounted for. So what was really going on? Leonard Hayflick was a young biologist in the 1960s who, in his paper titled The Limited In Vitro Lifetime of Human Diploid Cell Strains, challenged the notion that cells could divide indefinitely. In the paper, he outlined evidence that human fibroblasts in culture could only divide a maximum of 50 cell divisions, after which time cell division ceased to occur and cells died, a phenomenon later known as the Hayflick limit. This maximum division limit was fairly predictable, plus or minus 10 divisions. Even after freezing the cells at minus 80 Celsius, then regrowing them, cells remembered which division they left off at. These observations led Hayflick to propose the presence of a molecular clock that influenced the number of divisions. It took many years for the scientific community to finally accept Hayflick's conclusion that cells in culture are mortal. However, since that time, the idea that cells predictably stop dividing and undergo growth arrest has become the pillar of aging biology known as senescence. The molecular basis for why cells undergo senescence is now understood to some extent and can be broken down into replicative and premature senescence. Replicative senescence is an endogenous mechanism leading to senescence. It is defined by progressive shortening of what are called telomeres. On the ends of chromosomes, telomeres are composed of repeating DNA sequences that cap the ends of the chromosomes and protect it from DNA degradation. When chromosomes are replicated in preparation for cell division, the telomeres shorten with each successive division. This inability to replicate DNA, combined with the limited capacity for cells to add more telomere units, the cell refuses to divide. That is, unless you're a cancer cell, who loves to divide just a little too much. Premature senescence is caused through a variety of exogenous stressors. DNA damage, tumor suppressor gene loss, and exposure to excessive reactive oxygen species all play a role in pushing cells into senescence. Whether senescence is caused through exogenous or endogenous stressor, the result is the same. The cell undergoes cell cycle arrest, tumor suppressor gene activation, or DNA damage response. Some of these hallmarks of senescence can be observed visually, such as increased cell size and flatter morphology. The presence of beta-galactosidase, which is recognized as a key marker of cell senescence, can also be observed. In many ways, senescence is a good thing. Those cells that go on to replicate, despite considerable damage and mutations, may result in unregulated proliferation, also known as cancer. Therefore, it makes sense to employ internal mechanisms to shut down cell division, right? Well, it's a trade-off. Senescent cells are like zombies, not quite dead, but not quite alive. And interestingly, they have the capacity to turn nearby cells into more zombie cells by releasing pro-inflammatory molecules into their surroundings. This process is called senescence-associated secretory phenotype, or SASP. This bystander effect can lead to the very abnormalities senescence tries to avoid. This includes degenerative diseases like heart disease and cancer. While this might seem like a poor evolutionary trade-off, where the cost-benefit comparison between getting cancer and having cells senesced is negligible, this really isn't the case. Exercise and caloric restriction are associated with the clearance of senescent cells and the reduction in the aging phenotype. In addition, drugs called senolytics, which clear senescent cells in the body, are currently being researched to slow aging. Early experiments have shown promise in mouse models and clinical trials using drugs such as dasatinib, which is proposed to selectively clear senescent cells in the body. Clinical trials are currently underway in humans, and small pilot studies have shown promise. If this drug works, it may reduce the harmful secretory phenotype from senescent cells, keeping the surrounding cells happy, leading to less inflammation, less cell dysfunction, and hopefully slowing of the aging process. The full nature of cellular senescence is still in question, as is the nature of aging. As of now, the FDA does not recognize aging as a disease, which makes drug creation to reverse or slow it almost impossible. All the while, people slowly fall apart over time as our cells age and stop dividing. However, one day we may live in a world where senolytics are prescribed early in life to avoid the accumulation later in life of senescent cells. Hopefully, this will enable us to live not only longer but healthier lives in advanced age. While this is the end of my presentation, our understanding of senescence and cell aging is just beginning.